And I also thank Ben for giving a good introduction to coronaviruses. So this is the life cycle of coronaviruses, and particularly SARS-CoV-2, and the different drugs that are now in use just target different steps. For example, ribavirin is a broad spectrum antiviral that might inhibit capping in addition to many other things. And protease inhibitors, and also the remdesivir. The remdesivir actually inhibit the RNA polymerase, and also the NSP14 that we talked about just in the last talk, the proofreading enzyme. So for virologists, we always try to learn from HIV. So for HIV, for each of the steps of the HIV life cycle, we kind of have a specific drug that target each of these steps. And theoretically, for SARS-CoV-2, we might also do the same. For example, we might develop entry-blocking drugs. We might develop protease inhibitors, replicase inhibitors. And one specific thing is for the other enzymes, for example, this helicase. We actually have just developed helicase inhibitors. And in addition to these, there might be cellular targets, there might be inflammasome inhibitors that might prove useful. So in this recent study, we are trying to develop anti-SARS-CoV-2 drugs that target the helicase. So this is actually, we did this chemical screening, and then we got this small molecule, which is known as ranitidine, which is actually an anti-ulcer drug for high pyrrole. And as we shown here, that in these CACO2 cells, it can actually inhibit viral loads. And actually, if we compare the tissue viral load, we can also see that both in the nostril and the lung tissue, these ranitidine have actually can even did better than remdesivir in suppressing viral replication. And for the histology scores, we compare the nasal and lung tissue for inflammation. We can also see that these ranitidine actually have a favorable beneficial effect that can actually inhibit inflammation in the lung and in the nasal tissue. And when we perform a further biochemical study, we know that this ranitidine is a strong inhibitor of both the ATPase and also the dsDNA unwinding activity of the helicase. And we do kinetic studies and show that it is actually a good inhibitor. So these ranitidine does have the potential to be a new anti-SARS-CoV-2 drug that specifically inhibit helicase. And it is already in clinical use for high pyrrole, which is actually an anti-ulcer drug. So in addition to learn from HIV, another thing that we can learn is from the asymptomatic infection. So the SARS-CoV-2 and probably its parental virus, in their reservoir hosts, such as the bags, they actually are non-pathogenic. And also in a quite significant portion of humans, they are also asymptomatic. So how we could learn from the asymptomatic carriers, that's the question. So whether the viral loads are high or low in these asymptomatic carriers, or whether how often do these asymptomatic carriers shed a lot of virus? And what is the viral transmission from these asymptomatic and pre-symptomatic patients? And also whether in these asymptomatic infection, whether the people have strong and long-lived humoral and cellular immunity against the virus. So these are all important questions. And if we can learn more from these asymptomatic carriers, either in the animals like bats or in humans, then we might have a handle to understand the pathogenesis and also to develop new drugs that might be targeting the cell, for example. So what are the reasons for this asymptomatic infection? So maybe it's adaptation. So when these viruses have been well adapted in the reservoir hosts and in humans. So why they could be adapted in humans? One hypothesis is that maybe these SARS-CoV-2 might already been adapted in something which is very close to humans, for example, pangolin or some other species. So, and then the question is whether these asymptomatic carriers, they can mobilize a very strong antiviral immunity, and also whether they could actually have a weakened inflammation. And as in the case of HIV, the antiviral immunity, for example, the T cell response could even be decoupled from viral replication. And lastly, whether there is pre-existing immunity or cross-reactive immunity in these asymptomatic carriers. For example, whether they might have pre-existing antibodies against, for example, OC43 or HKU1 that are the community-acquired human coronaviruses. And there are some evidence in support of these, but still, the concrete evidence is still to be seen. So actually, this is actually similar not only to SARS-CoV-2, but also to HIV and even to influenza. So in their reservoir hosts, they are non-pathogenic, both in these chimpanzee and also in these sooty mangabe, HIV1, HIV2, they are non-pathogenic. But if they jump to the new host, for example, humans or other monkeys, they are pathogenic. So actually, Ben has already covered this interesting topic, whether the bats as the reservoir of human coronaviruses, why are they good reservoirs for these different human coronaviruses? You can cite high metabolic rate, high temperature, flying mammals. These bats are just long-lived and they just remain healthy always. And the two things that we know are, first, the high antiviral response, so they can mobilize a very strong interferon response. And then on the other hand, they have low inflammation, so the inflammation pathways are defective. And in these bags, there are a wide variety of different bags, and at the same time, there are a wide variety of different viruses. And this is actually through decades or centuries of co-evolution. So the viruses and the hosts, they are highly adapted and then non-pathogenic. So this is one of our recent study in 2014, in which we are actually comparing the MERS coronavirus interferon antagonist, the ORF4A protein, with, for example, this HKU4, which is the back homologue, very closely related to MERS coronavirus. And as we can see here, that actually the back virus, the HKU4, it could not bind RNA and it could not suppress interferon. So that's why in the backs, the interferon response are hyperactivated. But in humans, the MERS4A, it can bind RNA and it could actually inhibit interferon response. So this is one example that the viral factors could actually make a difference. So very similar proteins that are the interferon antagonists in humans, they could not inhibit interferon in backs, in back cells and in back organoids. And on the other hand, the cellular factors could also contribute to these asymptomatic infection. So in this study that we published last year, we showed that actually the SARS coronavirus ORF3A, it is a potent activator of the NLRP3 inflammasome through this mechanism that it can promote the ASC ubiquitination. However, the same mechanism is actually completely absent in back cells and back tissues. So that's shown by our friend Linfa Wang now in Singapore. So the documentation of the asymptomatic virus shedding is actually made very early on in early January by my colleagues, Professor K.Y. Yun. And this is actually the first paper, the first research article on SARS-CoV-2 and also in COVID-19, which document hard evidence for human-human transmission, high transmissibility with 83 attack rate, and also asymptomatic virus shedding, also diarrhea in some patients. And these, as you can see here, actually this study was done in early January. So I have high respect for Professor Yun and his colleagues because this is actually during the period in which the virus and the infection is still not acknowledged. And actually in our hospital in Shenzhen, the University of Hong Kong has a hospital in Shenzhen. They took the courage to admit the first patient actually on January the 10th. And then they chased the whole family and actually document all these with all the key features of SARS-CoV-2. And it is actually due to this hard evidence that he obtained within a very short period of time, within days. On January 17th, we actually managed to have these, to have Beijing change their whole control strategy. And it is this group led by Professor Yun, they actually truly make a difference in China. And this also is published quickly at the end of January. So also make the foundation for our understanding of the SARS-CoV-2. And in this follow-up study, we actually show in China, right after that, on January 7th, we actually showed that the asymptomatic carriers, they could pass on the virus to others. And this is actually the students who actually returned from Wuhan, and then they pass on the virus to the whole family. And again, our clinical colleagues in Professor Yun's group, this time Dr. Kelvin To, he documented the first case of reinfection in Hong Kong and also in the world. And in this reinfection case, first of all, the virus is different. There are 44 nucleotide difference, and that's about three and more months later. So the person was infected by another isolate, another strain of the virus. And as you can see here, the CT number keep going down. Keep going up, which is actually meaning less virus. And then the antibody comes up at day 5. So these in this reinfection case, the reinfection is actually asymptomatic. So that is to say the pre-existing immunity from another strain, which is 44 nucleotide different, still also it could not completely prevent reinfection. It could prevent disease. There is no symptom in this young man. So concerning the different strains, so whether the mutation rate of SARS-CoV-2 is significantly different, or whether it could actually make a difference so that it could actually escape immunity. So in fact, the mutation rate of SARS-CoV-2 is much less than HIV or even SARS-CoV-2. It's like only 50% of that of influenza and 25% of that of HIV. And that means that the SARS-CoV-2 might be well adapted to humans, and also the NSP14, the proofreading acid-ribonuclease, might also make a difference. But in addition to point mutations, our colleagues also found some deletions. And one is at the S1, S2 junction concerning the polybasic cleavage site for furin. And another one is actually in the ORFA, a few hundred nucleotide deletion. And importantly, both of these deletion mutants, they are attenuated in cell culture and also in the hamster model. And in addition to these deletion mutant, there are point mutations. The famous one, D614G. And whether the D614G mutation might actually make a difference. Indeed, it does have increased transmission, but no impact on pathogenicity. It loosens the spike protein a bit so that it can actually bind antibodies better, but still there's no escape from antibody. So these are the studies led by my colleagues Hongling Chen and Ziwei Chen. They actually have detected the deletion mutants initially at cultured viral cells and later on in COVID-19 patients, in which these polybasic site at the S1, S2 junction of the spike protein was found to be deleted, and the virus was found to be attenuated. So it remains to be seen whether some of these deletion mutants might become dominant. Now we don't have the evidence that they have already become dominant, but we should keep check on that in the surveillance system. So because in the backs we know that the interferon response is particularly strong, and there is no inflammation activation. So based on this reasoning, we actually can find that the interferon and also the IL-6 inhibitors, they might have beneficial effects. And indeed, my colleague also in collaboration with Professor K.Y. Yun, my colleague Ivan Hong, he show that the combination of interferon together with the protease inhibitor and ribavirin can actually have beneficial effect in hospitalized COVID-19 patients. And my friend in Hefei, Professor Haimin Wei, he showed that these IL-6 inhibitor also have beneficial effects. So to summarize this first part, the asymptomatic infection with SARS-CoV-2 is common in humans, and the mechanism is still not clear. And both viral factors and host factors might be involved. And the antiviral response and the inhibition, the activation of the interferon response and the inhibition of inflammation might actually be influential and could make a difference. And the pre-existing antibody and T-cell response might also play an important role. So for treatment of COVID-19, the repurposed drugs, the Taiwan interferon, ribavirin, protease inhibitor, and then the remdesivir, and then the steroid, and then the convalescent serum or the antibodies such as these regen COVID-2. But are the steroids doing more good than harm? So we need to strike a balance between the antiviral effect and the anti-inflammatory effects. The lessons from SARS and MERS told us that the use of corticosteroids does carry a risk. So this is actually whether to add fuel to the fire or to pull the button. So in the Chinese saying, as there are also Chinese adwords saying whether you are adding oil to the fire. So we need to strike a balance. So actually, in these very, very old studies in 1964 and 1969 on mouse hepatitis virus, it was found that the dexamethasone, also in very, very small amount, can already sensitize the macrophages to infection, and also the animal can change the genetic resistance in the animal. So with this in mind, we actually trying methylprednisolone, the infection of human macrophages from 3 different donors. And then we treat, we do pre-treatment, and we actually treat at 1 day or later.
Yang, you have 2 minutes.
Okay.
2 minutes.
I'll be fast. And then we can actually see beneficial effect for this methylprednisolone monotherapy, and the inflammation has been relieved. And then if we have methylprednisolone plus remdesivir, one important difference is actually at 14 days after infection, the viral load is lower instead of higher if we have methylprednisolone together with remdesivir. So that makes a difference. And the viral protein expression is the same. So if we have the combination, there's no expression. If we have steroid alone, we still have M protein expression significantly in the lung tissue. So to summarize these, the methylprednisolone sensitized human macrophages in infection, and the steroid reduced tissue inflammation, but could prolong viral clearance. But if we have a combination of methylprednisolone and remdesivir, it can have similar suppressive effect to inflammation, but also reduce the viral load. So Trump also have these remdesivir together with dexamethasone. So hope that actually would actually be helpful. So many people are now saying that that's Trump's steroid. So these are our collaborators and the people who are in my group, and the work, the the combination therapy work is mainly done in the hamster by Dr. Ye here. So, and these are the our team at Hong Kong U, led by Professor Yun, the different PIs in these state key lab. So thank you for your attention, and I'll just take questions.
Thank you, Dong Yan. And I'm going to go ahead and ask a question about the first topic that you discussed about ranitidine. And I wanted to know, you had mentioned that it was being used clinically to treat some particular disease. I didn't catch what that was.
It's the ulcer, the high pyrali, the bacteria.
Yeah, okay. And can you comment on, does it also inhibit cellular helicases? And
so it's quite specific for viral helicase. So that's we did a chemical screen, and the toxicity is much lower.
And in terms of patient delivery, how can this drug be delivered? Is it intravenous or is it a pill that you can swallow?
Intravenous would be better.